PLC machines. And this is kind of what you get. This is like, you get different fractions, and you're moving. Has anyone done chromatography in chemistry? So these are the early eluting molecules, the smaller molecular weights, and these are the later ones. And so we generate a lot of these diagrams, and he keeps sending them to me, and I actually don't know what they are. But what they are different tannins, basically. These are different phenolics, and so you can line up these different groups and quantify them and figure out what they are using mass spectroscopy and NMR. And I'm going to skip through some of this to get to the punchline, and that is that the white sand and the clay populations have very different chemistries, even though they're really recently evolved. This molecule here called oxidized terpene, where there's 2 or 3 times more being produced in the white sand than in the clay. Whereas this molecule here, this quinic acid derivative, is only being produced in clay and not being produced at all in white sand. Flavones are more produced in white sand than in clay. And this flavan is all the tannins. And we just sort of quantified all of them for this paper. But we've since analyzed them in more detail, and we found that although they both are investing a lot in flavans, the types of flavans are completely different in the two different species or ecotypes, whatever you want to call them. So we found completely different chemicals in the white sand versus the clay populations. And not only that, there were more of them in the white sand. So there were more chemicals and different types, and that matched some of the insect data that we found. There were more insects attacking the clay guys. And I think that this has a lot of potential as we learn more about the chemistry and more about the insects in this group. Because we have this phylogeny, we know a little bit about the evolutionary history of these groups across South America. Like I told you, there was this population here had that quinic acid derivative that the white sand ones didn't have. And so what if that was evolved here, in the history, and we went and sampled across the range and we didn't find that chemical? This new chemical that comes around could have something to do with the switch to these fertile soils in the western Amazon. It could have something to do with those herbivores that are attacking it. And we can go back now, we have a data set where we know something about what are the most common and dominant herbivores. We could take this molecule, we could do experiments with the herbivore and see whether or not it's toxic, or it's able to eat it, or it's like spicy food for it. These are things that we can do now that we know a little bit about the players in the coevolutionary story. So I'm pretty excited about that. All right, so I'm just going to tell you, I got like 3 more minutes, and I'm going to tell you just a few things that kind of the directions I want to go going forward. So the way that I think about how does herbivores promote habitat specialization, this is kind of like the way I think about it. Different traits are selected for the different environments, right? You have nutrient-poor soils and nutrient-rich soils, and the impact of herbivory is different in the different environments. So you have different traits are being selected for. And those are trade-offs, right? The advantage in one habitat comes at a disadvantage in the other habitat. And the same thing goes for this qualitative selection, where you have different herbivores are selected for different chemicals. If you have a chemical that's only going to function against a certain type of herbivore, but that herbivore doesn't exist in the other habitat, investing in that is going to be at a disadvantage. You're investing in something that is not giving you any benefit. And so these trade-offs are going to are basically causing these plants to become very locally adapted. And you can imagine if you're really locally adapted and you have gene flow with a neighboring population that's also locally adapted, what are you going to produce? You're going to produce intermediate phenotypes. Those intermediate phenotypes, they're not going to be locally adapted to either environment. They're going to be kind of intermediate. And my hypothesis is that intermediates are not going to fare well. They're either going to be easier to eat, or they're not going to compete very well against other plants that are more locally adapted. And so that could cause selection for post-zygotic isolation, to cause You know, if plants are creating these intermediate phenotypes that are never going to survive, then that's just wasting gametes. It's wasting energy, and so that's going to drive selection for incompatibility mechanisms. Okay, and that's kind of what my graduate student Tracy Misewich is studying. And so she's gone around and done like hand pollination experiments in these trees by climbing in. And these flowers, by the way, are just a couple millimeters in diameter. So it's a lot of work that she's been able to pull it off, to take pollen from one and save it and climb another tree and stick it in there and see what happens. And what she's found is that actually you can get cross-pollination to work. You can get hybrids to form, but they don't seem to survive at a very high frequency. So there's probably some kind of compatibility that's not complete, it's partial. And so that's a pretty exciting avenue of research that we'll pursue. If we can get the actual hybrids to form, then we'll be able to do all kinds of experiments with the hybrid seedlings. But that might not be possible. The other thing is, taking it out of scale, is I'm interested in this whole group, and there's more than 100 species in this group. They live mostly in South America, but also in different places in the tropics, in the Old World, in the Caribbean and in Central America. But we've also been able to map habitat onto here and ask the question at a really larger scale: how many times does habitat specialization evolve? And you know, this group has got fossils in it too, so we'll be able to kind of calibrate and figure out how long ago these speciation events happened, to what extent are they coincide with different geological events, like the collision of North and South America or the Andean uplift. And so I think that will be a very useful tool to have that phylogeny. And what we're doing now is doing that same kind of insect survey work and that same kind of chemistry work, but not only on that one species with the 2 morphotypes. We're trying to do it on between 30 and 50 species in this group. And we're also going to be trying to study the fungal pathogens at the same time, because those can kill a lot of plants also. And the idea is to know something about the enemies and who's killing these plants in the different places, and learn about what are the chemical defenses the plants have, and try to put all these data together to try to understand to what extent you have host specialists, enemies that are attacking these plants, the plants are defending them, like that co-evolutionary arms race hypothesis, or whether that's not the case at all, and most insects that are attacking them are generalists and eat a lot of different plants, and that maybe there are kind of other sorts of controls that are influencing plant chemical diversity, plant species diversity, and the word diversity. But my suspicion is that even though you don't have a lot of abiotic heterogeneity in terms of habitats in the Amazon, you have enough soil heterogeneity combined with all these insects and all these enemies. And those two things together mean you have more habitats, more environments that plants can become locally adapted to. And that might explain why you get this really strong gradient in species richness. What I'm showing you here is productivity. It's like how much biomass is produced per year on Earth. And so green is the highest and red is the lowest, right? And when I see productivity, I think of plant production, but I think of that as food for enemies. So this means how much food is there out there for things eating plants, okay? And then the things that are eating the plants. And so the tropics are where you have the highest intensity. And if those are interacting with edaphic gradients and environmental gradients, that may explain something about why you get high turnover in species composition and high regional diversity of plants. So thanks very much for your attention, and thank all the people that have helped me along the way, funding agencies, et cetera. And I'll be happy to answer any questions.